Hello everyone, and welcome back to MCAT Biochemistry. In today's lesson, we'll be discussing amino acid-base chemistry, one of the highest yield topics in MCAT Biochem. Understanding how amino acids behave in different pH environments is essential because these concepts appear in questions involving amino acid charge, protein structure, and many other various types. By the end of this lesson, you should be able to determine the charge of an amino acid at any pH, interpret amino acid titration curves, explain the significance of pKa, and calculate the isoelectric point. Let's begin. Every amino acid contains at least two ionizable functional groups, an amino group and a carboxyl group. These functional groups can either gain or lose protons, depending on the pH of the surrounding environment. As a result, the overall charge of an amino acid changes as the pH changes. According to the Lowry definition, an acid is a proton donor, while a base is a proton acceptor. Amino acids are capable of acting as both acids and bases because they contain both types of functional groups. This behavior is extremely important in biology because changes in pH can alter protein structure and enzyme function by changing the protonation state of amino acid side chains. Let's first consider what happens under strongly acidic conditions. At low pH, hydrogen ions are abundant. Because protons are readily available, functional groups tend to remain protonated. In contrast, under basic conditions, hydroxide ions remove protons from functional groups, causing them to be deprotonated. A simple way to remember this is low pH equals protonated, and high pH equals deprotonated. Nearly every amino acid-base protein on the MCAT is based on this principle. To understand how amino acids change with pH, let's examine glycine. Glycine is the simplest amino acid because its side chain consists of only an hydrogen atom. Under strongly acidic conditions, the amino group exists as NH3+, while carboxyl group remains COOH. The amino group contributes a positive charge while the carboxyl group is neutral, giving glycine an overall charge of +1. As the pH increases, the carboxyl group loses its proton first because it is a more acidic functional group. The molecule now contains both a positively charged amino group and a negatively charged carboxylic group. This form is known as a Switzer Eilon, and its overall charge is 0. As the pH continues to increase, the amino acid also loses its proton, forming NH2. At this point, glycine has an overall charge of -1. Notice the overall trend? As pH increases, amino acids become progressively less protonated and more negatively charged. Now, let's discuss pKa. The pKa of a functional group describes its tendency to donate a proton. A lower pKa corresponds to a stronger acid because the functional group gives up its proton more readily. A higher pKa indicates a weaker acid that holds onto its proton more tightly. Perhaps the most important relationship to remember is this. When pH equals pKa, the functional group is 50% protonated and 50% deprotonated. This point is known as the half equivalence point, and it corresponds to the center of buffering region on a titration curve. Now, let's apply these concepts to the amino acid titration curves. During a titration, a strong base is gradually added to an amino acid solution. The x-axis represents the amount of bases added, while the y-axis represents the pH. Initially, the amino acid is fully protonated. As the base is added, the carboxyl group loses its proton first because it has a lower pKa. The relatively flat region of the curve surrounding the pKa is called the buffering region, where the solution resists changes in pH because both protonated and deprotonated forms are present. At the midpoint of this buffering region, the pH equals the pKa. As additional base is added, the titration reaches the first equivalence point, where the carboxyl group has almost been completely deprotonated. At this stage, the amino acid exists predominantly as the Switzer island. Further addition of base causes the amino group to deprotonate, creating a second buffering region, followed by a second equivalence point. Eventually, both ionizable groups are deprotonated, and the amino acid carries a net charge of -1. Another important concept is the isoelectric point, abbreviated Pi. The isoelectric point is defined as the pH at which an amino acid has an overall net charge of 0. For amino acids that do not contain ionizable side chains, such as glycine and alanine, this corresponds to the Switzer ionic form. So far, we've focused on amino acids that only contain 2 ionizable groups. However, acidic amino acids, such as aspartate, contain an additional carboxylic side chain. Basic amino acids, such as lysine, contain an initial amino side chain. Because these side chains can also gain or lose protons, these amino acids have 3 pKa values instead of 2. This results in more complex titration curves. For acidic amino acids, calculate the Pi by averaging the 2 lowest pKa values. For basic amino acids, calculate the Pi by averaging the 2 lowest pKa values. Before we finish, let's review the most important points. Amino acids can act both as acids and bases because they contain ionizable functional groups. At low pH, amino acids are predominantly protonated, while at high pHs, they're deprotonated. Lower pKa values correspond to stronger acids, and when the pH equals the pKa, the functional group is 50% protonated and the other 50% deprotonated. Thank you for watching. If you found this video helpful, be sure to subscribe for more MCAT Biochemistry Explained videos.